Okay, cool. All right, should we get going? It's 3 o'clock. It's Saturday. You've all got places to be. And the last thing you want to be doing is listening to me. All right, so my name's James. I'm a colorectal surgeon. I'm a reader in surgery. And my group studies the oral microbiome. Well, it doesn't study the oral microbiome, it does a bit. But we study the microbiome in cancer, predominantly cancer causation. We are very interested in engineering the microbiome to improve efficacy and reduce toxicity to chemotherapy. And increasingly, my group is studying the early life microbiome. So today, I was in theatre today, and I spent my morning rather hurriedly trying to cut out a rectal cancer from a man in his 50s. If you're a millennial, your risk of bowel cancer is now 4 times that, the baby boomer generation, and it's climbing still in Gen Z, and Gen A will have an even higher risk. And the question I'm asking myself is why? Why is this happening? And if you want to understand the answer to that question, what you have to understand is that that is happening in context. It is not happening in isolation. The rising burden of non-communicable disease that we're experiencing is, of course, happening in cancer, but it's quite often happening in immune-mediated diseases like rheumatoid arthritis or inflammatory bowel disease. It's happening in different parts of the immune system, such as allergy, asthma. But we're also seeing a rise in non-communicable disease risk like autism, and we'll talk about that. So these are some of my disclosures. I sit on some advisory boards for some microbiome companies like Intus Biosciences. So this is what I'm going to talk about. I am going to talk about the oral microbiome because there's a renaissance and a great interest in the oral microbiome at the moment. But really, if you leave nothing from today's talk, I want you to understand the importance of the microbiome in early life and how it sets our risk of non-communicable disease throughout our life course. And I'm going to explain to you this concept of orchestral signaling, and I'm going to give you some examples of the things that we're studying, and I'm going to try and link it to the oral microbiome where possible, because I believe that's where your core interest lies. So this is the problem. This is data going back from the 1900s. What you can see, this is daily adjusted life years, and it's things that cause us problems. And what you can see is respiratory infections and tuberculosis, and things that killed us because they were pathogenic, infectious diseases effectively, have declined, and they've declined rapidly. Neonatal survival, it improves, and you can see cardiovascular disease is just, it is the disease of our lifetime, isn't it? 16% of the world's population die of a stroke. The majority of us will die from cardiovascular diseases. I've had my CT angiogram. My right main stem is 20% stenosed. So, I'm just waiting for it, basically. But, the microbiome is important because it allows you to change it. What's interesting, I think, also in that is the rise of mental health diseases, which, of course, are multifactorial. It's not all related to the microbiome, but the microbiome, I believe, is part of it. And the basic essence is we are living longer, but we are not living happier. We are living with this burden of disease, and this burden of disease is not fairly or equally distributed. It is young people that unfairly carry the burden of it, and of course we have an aging population who are also going to carry an extreme burden of this. Now, you've probably had lots of brilliant speakers today. I'm really, really sorry I wasn't here for the beginning of the day, so forgive me if anything that I'm going to tell you is repeated. But I think these things are worth repeating. In all of the definitions that you've been given of the microbiome today, there are 2 fundamental things which are the most important. The first is that we have an evolutionary relationship with it. And we have co-evolved. We haven't evolved in isolation, and sometimes that is described as a phylosymbiotic evolution. Microbes have shaped our evolution, sometimes profoundly and fundamentally, and sometimes they've just been happy passengers as we've sort of separated off into our individual branches. And the microbes that are in your mouth, and the microbes that are in our gut, and the microbes that are on our skin, and the microbes that are in our urogenital organs are not there by accident. They are there for a very important reason, and they are there, and they've been there, by the way, for millions of years, and they will be there probably long after we've gone. So this is data looking at dental plaques from Neanderthals and comparing it to chimpanzees and to the modern human. And this is data at a fairly kind of broad descriptive level, taxonomic level. And I think there are 2 kind of key things. Generally, it's perceived that in the last 10,000 years, we've probably lost about 40% of the human microbiome in terms of its diversity. And you can see the Neanderthal was kind of, its mouth was dominated by actinobacteria, and that was probably representative of its diet and how it's lived its life. And you can see that those have almost completely disappeared from the modern mouth, and they're still quite dominant in the chimpanzee. But what's interesting about microbial evolutionary biology is, like any other aspect of evolutionary biology, there are winners and there are losers, right? And in the modern mouth, Bacteroidetes, bloom, right? So because bacteria can rapidly transfer gene mutations, horizontally, and they can share those, they are tremendously good at surviving in an austere environment, and the mouth is an austere environment. It's, we have lots of extremophiles in the mouth, and we'll talk about some of them, right? So there are winners and there are losers in modern life. And in fact, there seems to be kind of big events that have had a huge impact in shaping our microbiome. So what this data shows is this quite, I think this work might have even been done at UCL. If it wasn't UCL, it was King's. And this was, again, an evolutionary biologist that was looking at the evolution of the human microbiome and the oral microbiome over years. And there seems to have been major events in our history, and the Black Death was one of them. So in about the, God, I forget, I can't remember if I put the date down on this. So it was, was it 410 CE? Anyway, you can see down on the graph here, I can't quite see it. After the Black Death, there was a mass culling of the oral microbiome. It seemed to fundamentally disappear. And that might be for a number of different reasons. It might be because our society changed as a net result of it. It might be because of the direct relationship between pathogens and between the symbionts that live in our mouth. Anyway, my point is there have been important events throughout our history which have had a very big impact on the microbiome. Now, we are definitely experiencing, well, I don't want to be too controversial, but many scientists believe we're experiencing a mass extinction event currently, right, globally. We are experiencing a global loss of the biodiversity of our ecosystems, and humans are no different. Within us, we are also experiencing a climate crisis, is how I think about it, or a mass extinction event. And that's been driven in part by these heroes, right, these scientists who you will all know, who have saved millions and millions and millions of lives because they understood the mechanisms of the majority of infectious diseases. They discovered the cause of things like tuberculosis. They have shaped the course of humankind through the development of antimicrobial, antisepsis strategies. And what do you notice about this picture that I'm showing you? Does anybody notice anything about this without wishing to be too woke? They're all men. They're all men. They're not very diverse, right, are they? Biodiversity is a good thing. Diversity in our scientific organizations is a good thing. There was no diversity. Again, I'm slightly preaching to the converted, but, there's a big gap in a lot of microbiome sciences around ethnic subgroups, African subgroups. We're not very good at studying important cohorts of patients that really deserve to be supported and studied. Anyway, no women in that picture. There was one woman. This is Margaret Jennings. She did all the toxicity work for penicillin, right? So she was obviously very, very important, completely forgotten from the history books. But look her up. She was important. She married Howard, what's his name? I want to say Florin, but it wasn't. The guy that won the Nobel Prize. She was married to him anyway. So basically, germ theory has shaped the human microbiome. It has, in fact, shaped the planetary microbiome, I would argue, and I will talk about that in a little bit more detail. So my basic hypothesis, however, is that the rising rates of diseases that I have to treat, like bowel cancer, are a side effect of this internal climate crisis, in this internal loss of biodiverse ecosystems. So your microbiome has an evolutionary basis. The second thing that's really, really important is that it has a time-specific basis. Your microbiome is not stable with you throughout your whole life. It is very, very dynamic in your early development phases, so up until about the age of 5. Its structure, its ecological structure, its metataxonomy is locked in about the age of 5 years old. But then its functions continue to adapt, right, until about puberty. Then it really stabilizes. And in adult life, it's pretty stable, right? All your microbiomes are pretty stable unless you have a disease, surgery, something catastrophic or really fundamental changes in your life, until you get to old age, and then it becomes unstable again, which is a problem for me because many of the patients that I treat are old. The third, I said there were two, but I'm going to chuck in a third. The third thing is that it's very, very individualized. So your microbiome and my microbiome might share similar core genomic functions, but at species and strain level, it's very, very different. So this is 174 of my patients expressed at phyla level. This is the mucosal microbiomes of patients with bowel cancer. And what you can see is just they're very variable in that cohort of 174 people. What does that mean? I don't know the answer to that question, either in terms of causation of cancer, progression of cancer, or therapeutic response to cancer, but that's the question we're trying to answer. And what is the value of that variation in terms of the way that we treat cancer? I think it's very important, is my personal belief, but we'll talk about it. So, brilliant film. If you've never seen it, it's really worth another watch. Goodfellas, you know, Martin Scorsese, classic. And this is a quote from Henry Hill about the mob, right? And it could be talking about probiotics in the gut, frankly, okay? There is a lot of bullshit spoken about the microbiome. And largely that bullshit is put forward by probiotic companies mis-selling probiotics. There is no such thing as a good or a bad bacteria. This is nonsense. There are pathogens and there are symbionts. And if you treat your symbionts badly enough, they will bite you and you will have a non-communicable disease, okay? So actually, it's about gene, environment, microbiome interactions, and it's about how you nourish, nurture, and care for your microbial and your internal ecology, and that's how you maintain your health. And that requires a philosophical shift. It requires an evolution from germ theory into something that is more nuanced. So I just put this picture up really because it's pretty. So this is a dental plaque. And this is something called classy fish. It's a way of immunostaining particular strains of bacteria with a fluorescing biomarker. And you can see the nuance and the blooms of these microbes within a single dental plaque, right? And it's very pretty. But actually, it's very nuanced. And what it tells you is that bacteria and microbes do not exist in isolation. Although we think about them in that 19th century way as single pathogens, because that's how clinicians often were taught to think about it, that's not what's happening in practice. They're communicating. And some of those are in discrete niches, isolated on their own, and some of them are communicating across whole networks. And that is very challenging if you're trying to study it, to try and understand mechanism and disease causation, because it requires complexity, right? And a nuanced understanding of that complexity. You know, that's 100 microns across, by the way, at the top. So you can suddenly get a sense of the kind of overwhelming scale of it. And that nuance exists in the oral microbiome because the oral microbiome is not a homogeneous entity. The microbes that are on the dorsum of your tongue are going to be very, very different to those that are going to be in a dental plaque. And even when you look within particular species of bug, or taxa of bug, like streptococci, and you look within different regions of the mouth, there are going to be very different types of microbes there. And that is important. It's important if you're trying to understand the microbiome, and it's important if you're trying to give microbiome-targeted therapy. So I often treat a disease called inflammatory bowel disease. Many of you may know people with inflammatory bowel disease. Many of you may even have inflammatory bowel disease because it's disappointingly and worryingly common. And what we know is that the rates of IBD are climbing globally, but they're not climbing in a fair way, and they're climbing in countries that are being urbanized and are being modernized the fastest because it's a westernized disease. And what we've always known about the microbiome, and so what we've always known about IBD is that it's a product of gene-environment interactions. There's some people who are genetically susceptible, you dump them in the wrong environment, their immune system can't handle it, and they develop IBD disease. My hypothesis is that the microbiome is a player in all of this. So what this graph shows is basically my theory of everything. This is my theory of everything. And it may not be right. We can argue about it. So I believe there are three fundamental forces that cause non-communicable disease. There's your genome, there is your exposome, the environmental drivers of disease, and then there is the microbiome. I believe the microbiome I believe it's that profound. It's on that level. It's on that scale. This is my personal opinion. And what I've said to you is that you have a time dynamic relationship with that microbiome. And in fact, that microbiome starts planning influencing your health from the minute of conception, has a big role to play in infertility, in fact. But actually, the maternal microbiome signals to the gestating infant from the minute of conception. And then it's pretty, by the age of 3 to 5, maybe, it's pretty fixed. And I've mentioned it's pretty stable until you hit your 60s and 70s, and then you die. At some point, you go on a different trajectory and you get a disease. Something happens. And the question is, can you predict at which point that's going to happen? And can you predict in whom it's going to happen? And the answer is maybe. But I believe that if you want to be able to predict it accurately, you've got to have a measure of all three of those things, the exposome, the microbiome, and the genome. And what I'm arguing, and what others are arguing, Martin Blazer argues this, he wrote a good book called Missing Microbes, I recommend reading it, is that effectively this line, this gene environment microbiome line, is shifting. And it's shifting because it changes with each generation. So the Gen Z population have a very different microbiome, and they have a very different exposome to that of the post-war generation. And they're a hyper-global generation. And their microbiome lacks resilience. And their immune systems are therefore not educated in the right way. And that leads to the high risk of inflammatory bowel disease. And we can measure this. And we can measure this longitudinally in high-risk populations. So these are data from patients' families with inflammatory bowel disease that have been studied longitudinally over the duration of their life course. And what you see is that, I don't know if I have my pointer will show here, in this graph here, when you look at fairly simple measures of metataxonomy, is that you see changes in the microbiome long before the disease manifests itself. And that families with IBD share particular niches of these microbiomes. And that's because they have similar environmental exposures and they share their microbes, right? What you're seeing in microbiome science is a sort of move away from what has really been the first 15 years of microbiome research, which was basically association studies. It was a sort of an avalanche of association studies, and I can't be too critical because I published some of them. And that was fine and it was exciting, but it's not enough. It's just not enough. And what we initially thought was, well, because we're all 19th century germ theory thinkers, that actually there must just be too much of the bad thing. It's too many pathogens, it's too many pathobionts. And if you look at a disease like inflammatory bowel disease, we've got a list of bad actors that we know are consistently and serially overrepresented in patients with this disease. And then we thought, well, it might be loss of symbionts. Maybe it's not enough of the right bugs, you're losing them. And we've got a list of those that seem to also be quite important. But when you try and apply them longitudinally in study populations, these models are not effective predictors if you just look at individual strains of bugs, because they're only part of the story and because it doesn't explain mechanism. You have to move away from simply a bug being present to be a biomarker of disease to really understanding how it's causing a disease. And that's where we are now with the microbiome. And the microbiome, by the way, is not just bacteria. Like, I can't stress that enough. I'm going to talk about bacteria because we've got to have some sort of description of what's going on. The microbiome is very much dominated by viruses. It is very much dominated by phage in particular. Low abundance members of the microbiome, like fungi, play a very important part in shaping the microbiome. Eukaryotes in the gut also, have a very important part to play. 2 billion people around the world have endemic worms in their gut, and these are very, very important. So what we're moving towards is microbiome host dynamics, right? And epigenetic programming would be a really good example of that. How bugs in the gut reprogram genes and how that causes and drives disease, for example. And we're getting into the detail of that. But nonetheless, there have been a couple of consistently recognized bad actors that we come back to. And Fusobacterium nucleatum is probably the number one poster child for this. So in bowel cancer, where I work, over the last sort of 5-10 years, what we've been trying to do is take those bad actors and work out how they might cause genomic instability and then cause and drive cancer progression after it's initiated. And again, there are a couple of other bad actors that we're identifying. But fusos are interesting because they belong in the mouth. That's where they live. And they turn up absolutely everywhere. They turn up in breast cancers, pancreatic cancers, gastric cancers, bowel cancers, liver cancers, bladder cancers, cervical cancers. And the question is, well, how? How are they getting there? Why are they there? And what does it mean for cancer biology when it happens? So this was some data looking at patients with pancreatic cancer. So these patients, pancreatic cancer has appalling outcomes because it's usually detected really late. It's very, very aggressive and patients do very badly. And I want you to focus, because there's quite a lot of pictures here. My mouse, do we have a laser pointer? If you look at this figure H down at the bottom here, what that shows you is you go from IPMN is a kind of precursor lesion of pancreatic cancer, to low-grade dysplasia, to high-grade dysplasia. You start to see higher and higher and higher abundances of these oral bugs turn up in these precursor lesions of cancer. And we find that in other diseases. So bowel cancer, for example, when we've done studies trying to match specific strains of Fusobacterium nucleatum from the mouth onto precursor lesions of cancer, they correlate very highly, which means that they're the same specific strains that seem to be causing it. And this paper came out this year. Sadly, it wasn't my group. That explains part of that journey. And the main message from this slide is that precision in microbiome science is absolutely everything. So having a knowledge about a species just isn't enough. So what this data showed, that a specific clade of fusos, particularly Fusobacterium nucleatum animalium, seems to be a very discrete and important driver. And what those two figures show is that something happens in patients who've got bowel cancer, in that this very specific clade then is able to grow on and within these cancers. So cancer has an intratumoral microbiome. These microbes invade the cancer. And the question now, therefore, is can we develop and devise an oral biomarker for bowel cancer based on precise analysis and measurement of these? And probably more importantly, can we identify the moment that these oral microbiomes suddenly begin to engraft and grow in the gut? Because that's probably the moment where you really, really need to do something about it. What we found in our work, again, I'm sorry, there's lots of science in this, is that actually because we've studied in some of the patients that I showed you on that first slide, that actually it's a kind of nuanced, it's a nuanced picture. It's not as simple as saying there's too much of something and not enough of the other. Actually, what we find is discrete subclusters of these microbes that grow in these cancers, and quite often they're defined by tumor biology. They're defined by the genomics of that cancer. But they're very, very useful clinically. They're highly prognostic. They tell us who's going to survive and who's not going to survive. And to us, that's super interesting because we want to know why. So we've done other studies where we tried to identify these oral microbes in very discrete regions of the bowel. So we studied the right side of the colon because the right-sided cancer biology is very different to the left. But importantly, when we operate on these patients, we don't give them bowel preparations. We don't clean out their feces, which means that we can match fecal samples to mucosal samples to the cancer, and we can study the geography of this bowel. And what we find is that the molecules that these microbes produce is very variable across the geology of the gut, and that these microbes, they're very particular molecules that we're interested in called lipopolysaccharides, seem to be discretely secreted by very particular genus of bacteria, and these seem to particularly correlate with particular subtypes of cancer. In this case, cancers that are microsatellite unstable. That's important because microsatellite unstable cancers can be treated with immunotherapy. So we think you can target immunotherapy based on a measurement of microbial metabolism effectively in these cancers. And we'll come back to that, so hold that as a thought. I talked about inflammatory bowel disease. I've talked about cancer. If you have inflammatory bowel disease, your risk of bowel cancer goes up because you live with chronic inflammation in your gut. And in our group, we're asking why. Why does that happen? And we've gone hunting for these oral microbes in the gut again. And this is some work done by one of my PhD students, Duncan Roberts, who's just been awarded his PhD. We're very proud of him. And what he found was interesting. He found that these oral bugs behave very differently in sporadic cancer and these colitis-associated cancer. The Fusobacterium nucleatum animalium story doesn't hold. There's something very different happening here. Those microbes are still present, but they're not engrafting and they're not driving that cancer in the same way. Instead, we see enrichment of Escherichia and Shigella. Other groups around the world are beginning to explain how these microbes are driving cancer. And they seem to produce highly genotoxic small molecules, in this case indolamines, which seem to drive that mechanism. So the oral microbiome isn't just implicated in cancer and IBD, it's implicated in a whole host of different chronic diseases, and you've probably heard about them all today. And I'm not going to talk about them all in too much detail because we'll lose a lot of time. But I will talk about some of them specifically. This kind of blew my mind, actually. I found this, I was giving a talk to a society that treat children with cleft lips and cleft palates. And I found this study, and it was interesting to me because they were able to take saliva from patients with hypertension and do an oral salivary transplant into a mouse model and identify very specific species of Veillonella that could transmit a phenotype of hypertension. So and actually what they were then able to do is to identify single strain dynamics that are able to drive that. That sort of blew my mind, that an oral, salivary microbe can make your blood pressure go high. Maybe you know that as dentists. But in our work, what we are trying to understand is what are the big environmental drivers that are reshaping the microbiome that might explain some of these observations that we're seeing. And increasingly, I'm of the opinion that this is the most important thing, which is why I was showing you that picture of Fleming. And I work opposite Alexander Fleming's old laboratory, actually. I walk past it every day. Apparently a really grumpy man. Apparently. Didn't have much of a sense of humour, notoriously boring. But it didn't matter because he was doing good science. So we prescribe an enormous amount of antibiotics, nearly 35 billion daily doses of antibiotics a year. These antibiotics are not prescribed equally. They are predominantly prescribed to children. They're predominantly prescribed to people in lower middle income countries. They're majority prescribed in India and in China, where there's much less regulation around their use. And in fact, the majority are used in farming. 80% of them are used in farming. This is one of the original papers from 1946, published on the use of streptomycin in farming stock. So as we were coming out of the Second World War, we had a starving population and we couldn't feed them. And Pharma immediately understood that actually their biggest application might be in feeding the post-war population. So they were typically deployed in farming, used as basically an animal husbandry tool, so your animals didn't die. But they quickly realized that everybody's animals got fatter much faster. So you could get a chicken to market weight in probably a third of the time than you could without antibiotics. And then they became a drug of abuse. And they were systematically missold by pharma for decades, really until only very, very recently have we had legislation that stopped that. Our legislation for antibiotic misuse in farming in the UK is still well behind post-Brexit. So I find this interesting, right? I find this interesting that you can kill a bunch of bugs in a gut and the animal gets fatter, right? And if you go to China, they're still doing it on an epic and massive scale. If you go there, this is a 26-story pig skyscraper, right? Slaughters 1 million pigs a year, right? That takes a lot of antibiotics to do that, right? Now, some of us are very susceptible to antibiotics, and some of us are not. You probably know this from your clinical practice, right? But this was an interesting study that was published in Washington that I go back to time and time again. They took healthy individuals and they gave them a single dose of azithromycin. And they compared them to a cohort of patients that had been in intensive care. And then they followed them for 200 days to see what had happened. And what they found was that not only when you took a single dose of this broad-spectrum antibiotic that you had a 10,000-fold reduction in your antibiotic, in your microbiome abundances, but in some of those individuals, those abundances never really came back. So there are some of us that are just highly susceptible, and 200 days out of a single dose of antibiotic, their diversity was significantly less than the healthy control population. And in fact, it was so much perturbed that it resembled the cohort of ICU patients. But the thing that is more interesting to me is not the loss of diversity, which is a metric that has value. It's a loss of function. Almost every single function of the microbiome was changed with a single dose of antibiotic. And what we see is that antibiotics obviously influence infant health. So the oral microbiome, probably the best example I can give you is cleft palate. Gestating mothers given antibiotics in the first trimester, being treated for urinary tract infections with specific antibiotics have a higher risk of having children with cleft palates. We know from big epidemiological data studies that macrolide antibiotics, particularly prescribed in the first 3 months, increase almost all your risk of almost all malformations. But it's more nuanced than that, right? It's not just about malformations, because the majority of our children don't have malformations. They have obesity, they have asthma, they have allergy, they have a rising burden of mental health problems, right? So my hypothesis is, are we basically just treating our kids like those battery chickens from the 1940s? Why is it surprising that they're obese? And it's not. So if you look at these longitudinal studies of kids done in Finland, these were Finnish kids, I think between the ages of 6 and 7, and they followed them up. M6 is the moment where they had their macrolide antibiotics, and you can see they're all outliers on that kind of principal component analysis. And they all had increased risk of obesity. And we know from many epidemiological data sets that recurrent antibiotic use in childhood increases your risk of type 2 diabetes. It increases your risk of asthma. In fact, increases your risk of colonic adenoma formation and bowel cancer formation. The bigger question is, does it change your risk of mental health problems? Well, there's increasing interest in gut-brain interactions. And from the top, we have to, of course, put in the caveat that these things are multifactorial. You, I'm sure, are aware that TikTok probably does a lot of damage to our developing infant brains, but nonetheless, that is happening in a broader context, right? So I'm not saying that it's the only thing, but I'm saying it's a part of it. And what we're beginning to understand is the complexity and the nuance of that interaction and how it really works in practice. And the immune system seems to be like one of the most important determinants of brain function. And we're even beginning to unpick the detail of how the brain and the gut are hardwired together. So, for example, this paper was very, very recent. It was only two, 3 years ago. We discovered neuropod cells. So neuropod cells are sensory neurons in the small intestine that actually directly communicate with the luminal microbiome, with the luminal microbiota. And what's interesting is when you're stressed and when you're anxious, you can shape the microbiome. You change the ecology of the microbiome. Similarly, when your microbiome is unhappy, it has a direct and hardwired route straight into the brain to influence mood. And that might be also important in brain degeneration, for example. Okay, so Alzheimer's, for example, would be a really, really good example of that. We know from good fecal transplant studies in animals that when you transfect fecal samples from patients with Alzheimer's into those animal models will start to demonstrate signs and symptoms of Alzheimer's. And we know some of the mechanisms through which it happens. Short-chain fatty acid metabolism or co-metabolism would be a good example. And oral extremophiles are part of that narrative. They're part of that story. So Porphyromonas gingivalis, for example, like it's a non-motor gram-negative bug. I think of it as an extremophile that lives in the mouth. It's got this ability to adapt to a massive pH range, right, 5.5 to 8.5. And it can deal with very, very wide variances of oxygen tension and temperature deviations. And it produces toxic proteases. And what's quite interesting is that other microbes in the gut will produce biofilms and it will coalesce and live in those biofilms. So it has a synergistic relationship with other bugs that live in the mouth. And we're beginning to understand how it gets to the brain and how it produces vesicles to allow it to do that. But we're also beginning to understand the molecules that it produces, right? So it produces this protein called gingerpain. And gingerpain can be identified in patients that have Alzheimer's disease, and you can transfect that into an animal model, and you can trigger neurodegeneration in an animal model through Porphyromonas gingivalis administration in the mouth. What I also think is important, just worth 2 minutes of discussion, is this concept that actually the brain has this moment in its early development phase where you can interfere through the microbiome in its neuroplasticity and in its neurodevelopment. So fecal transplant studies of kids with autism spectrum condition, where you take feces from a kid with autism and you put it into a mouse, you can demonstrate autistic behaviors in that mouse through fecal transplant. And that is quite a consistent observation. But the most interesting thing to me is that only happens up to the point of weaning. After weaning, that effect is gone. You lose it. And the question is why? And the question is, what does that mean for our kids? Probably quite a lot. Kids with oral cleft defects have higher rates of neurodiversity. It's interesting to think that maybe they also have high rates of ASD, ADHD, and it's interesting to think the microbiome might be part of that story. Who knows? So, to finish, why are we experiencing this internal climate crisis? Well, yeah, it's antibiotics, but it's not just antibiotics, is it? You all know that. The majority of our global populations are urbanized. We have shrinking social networks. We live in increasingly smaller family size and family networks. We are exposed to more environmental contaminants. But this is kind of how I think about it, right? This is a kind of a timeline for what I think. If you want to understand why people are getting more bowel cancer, you want to understand why people are getting more IBD, you want to understand why more people are getting mental health problems, you have to think about those things in a time-dependent manner. So The maternal microbiome begins to shape health. Mums that have antibiotics, for example, if we take these data, have a risk of having children with Crohn's disease, which is 2.5 times that of the normal population. You have a baby, that maybe that baby doesn't have breastfeeding, maybe that baby is born into a world where its microbiome is not allowed to assemble robustly. Maybe it's hit with yet more antibiotics. Yes?
Is there a difference, or has it been studied yet, whether it's like a natural birth or a section birth?
Yeah, really important question. Has been studied a lot. Yes, it is quite a big difference. So the question is, cesarean section versus natural delivery, how does that change the microbiome? And the answer is it does have quite a big impact, because the microbiome is seeded through the delivery, through the birth canal, and it shapes it. But it's more nuanced, because children who are premature are more likely to have cesarean deliveries. Children who are cesarean delivered are more likely to have antibiotics or to be given antibiotics. So there's quite a lot of confounding in it. And I think probably prematurity is a bigger driver of it, actually.
And then I've heard
Yeah.
quite a few people discussing, like, kind of basically putting the vaginal microbiome
Yeah.
on the child for
Yeah.
them to swallow it. Is that?
Yep, that is also a thing, and so you can swab, exactly, vaginal mucosa, mucus onto the baby's mouth, and that is quite commonly done, and that does have an impact on the microbiome. But in a good way, yeah. But what I would say to any of you thinking about having children who are slightly disgusted by that is that what's much, much more important, I think, is that babies are sustainably breastfed for a longer period of time. So I think what's much more important is how you colonize the microbiome through the birth canal, but actually how you sustainably grow it through those kind of first 6 months of life. So when you breastfeed, what happens is you get a big super bloom of bifids that are driven by the galacto-oligosaccharides in breast milk. And then the breast milk is obviously a living tissue. It has its own kind of immune memory, which begins to shape the emerging and the developing microbiome. And what we also see is that kids who are breastfed and kids who are formula-fed have very, very different microbes, right? Formula-fed kids have Klebsiella and lots of these kind of pro-inflammatory microbes in their gut that are just very, very different. So I think the trouble with all of this is that pregnant women often are left to feel guilty or feel like, oh God, now I've made the wrong choice for my baby and all the rest of it. And it's kind of difficult. And my messaging is always, do what you think is right for you and your baby at that moment in time and follow the medical advice. But if there's one piece of advice I can give new families, it's breastfeed, breastfeed, breastfeed.
And is there, from your perspective, how long is breastfeeding encouraged?
So the WHO guidelines is to 6 months. But we reckon actually it should be, well, the advice is to you then introduce solid foods, but if you can breastfeed up to 2 years, that's what.
Is there any then gain benefit in the microbiome? Like you go to say 4 years, 5 years?
Well, not to the microbiome. You can argue that I'm not a breastfeeding expert, so you would need to really ask a breastfeeding expert. So I'd slightly plead the fifth on that. But I think that there are definitely health benefits up to 2 years. Whether those health benefits extend beyond that is another question. I don't know the data on it. But I also think it depends on where in the world you're breastfeeding. I think if you're breastfeeding in West London, you're probably all right to 6 months. I think if you're breastfeeding in, Yemen, it's a different ballgame. So hit 2, I think what happens is that you then have a gut that hasn't been assembled, that's been hit, that doesn't have optimal functions, and then it's hit with ultra-processed foods, it's hit with an exposome that contains a bunch of microplastics, maybe you smoke, and you've got a microbiome that just simply cannot metabolize it. It produces lots of secondary co-metabolic signaling molecules, and then at some point you have more antibiotics because that's what we like to do, and you flip into a disease state. When you develop a disease state, what happens is that the microbiome changes again. So the mucosal microbiome that we see on cancers does not necessarily reflect the microbiome that caused that cancer. The thing that caused it, the interaction, probably happened many, many years ago. So we talk about passengers and drivers, right? And the question is, are there oral microbes that we're measuring and we're detecting passengers, or are they drivers? We don't know the answer to that question. We're trying to work it out. So I was going to talk a little bit more, but I've talked quite a lot, and it's quite a lot to take on board. I do appreciate it. So I'm going to finish on this slide. So this is basically what I'm arguing for, is a shift away from germ theory. Not because it's wrong. Clearly, a lot of it was right and extremely important. And we still need to kill pathogens. And infectious diseases are still big global health problems, right? But actually, the bigger health problem that we've got globally is non-communicable disease. And what we need to do is to evolve to a microbiome theory, and that microbiome theory needs to really be about microbial conservationism. It's about instead of saying, how do I kill the microbes in my mouth that are going to cause periodontal disease or caries, whatever it is, how do I promote and enhance that microbiome, not just so that it prevents oral health problems, but actually how it prevents many other non-communicable diseases, which in fact may seemingly be unconnected to the mouth. The second bigger question is, how do we engineer these mutualistic microbes, and what does the future of that look like? So, does that mean taking a probiotic? Probably not. It probably might mean taking a next generation probiotic, which is targeted and really engineered to a specific function. Well, probably it means one of two things. Synthetically engineering microbes in a lab to really engineer them to do precisely what you want, or giving whole colonies of microbes through fecal transplant or oral microbiome transplant. I've got one slide on that. In fact, I'm going to talk about that. So so it's this. So we are now seeing the first oral transplants happen, where you're able to take healthy donor oral microbes and transplant them to someone who has, in this particular example, perioral disease. How you do that is surprisingly difficult. It's not as easy as taking a sample of saliva and just, snogging someone, as to Victoria's point earlier, actually. Because these microbes have to engraft. They have to grow. And they have to be nourished, and they have to be sustainably engrafted. So it's actually quite difficult. So in the examples that we've seen, actually, the recipient site needs to be prepared. And there is big debate about whether or not you should disinfect the mouth before you then transplant the mouth. We increasingly think you shouldn't. And this is all highly experimental and hasn't really been done in clinical practice yet. But I can totally see this one coming down the road. All right. So in summary, we're experiencing an internal climate crisis, I think. And by creating an extreme external climate, we are stressing our internal ecosystems. And that, in turn, I think, is causing this risk of non-communicable disease. And future strategies for oral health, therefore, must be targeted at improving the bioresilience of the mouth and improving the microbiome health through microbiome theory. And the best time to do this is unquestionably in early life and in old life. So that's it. Thank you very much for listening. Thank you. Thank you.
Thank you very much for a very interesting talk.
No worries.
You mentioned that the cancer tumors in colorectal cancer, they've got their own microbiome, which can contribute to disease progression.
Yeah.
Is that true of other cancerous tumors like in the pancreas as well, or is it just the colorectal?
It's a very interesting question. So it is true, but the bowel and the gut is a little different because it's got such a high burden or a relative abundance of microbes that are co-located with it. So it seems to be a more dominant feature of those cancers. What we understand is that these cancers, when they have these kind of intratumoral microbes, that it really changes the behavior of these cancers. So we see these microbes in metastatic deposits. We find them in liver metastases. We find them all over the body. And they also drive drug resistance. So we know that fusos, for example, play quite an important part in determining chemoresistance to therapy, which is why we're so interested in them. Increasingly, what we're interested in is actually not the bugs in the cancer. We're interested in the microbes around the cancer, right, in what we call the tumor microenvironment. And since you asked the question, that's because there's a new class of drugs. So the more modern class of drugs in cancer therapy target the immune system. There's a class of drugs called checkpoint inhibitors, which effectively allow T cells to identify cancer, and it allows the immune system basically to target the cancer. Fabulously expensive drugs, extraordinary drugs. Like, they are transforming outcomes from melanoma. We had a big trial published in rectal cancer last year. Patients with metastatic disease that were going to die suddenly just don't die. And it's amazing. It's absolutely amazing. But what we know is that the tumor microenvironment has an intimate and profound relationship with the microbiome. And the microbiome determines how the tumor microenvironment works. And that's why nutrition and diet is so important in cancer therapy, because diet-bug interactions change the tumor microenvironment. And it also means that the microbiome becomes a therapeutic target in its own right as an adjunct to cancer care. So this is an example of a paper that was published in Science about a couple of years ago. These were patients at Memorial Sloan Kettering, I think. They had advanced metastatic melanoma. They'd been on checkpoint inhibitors, and they became resistant to the drug, and they were dying, and they were going to die, and they had cancer everywhere, and that was that. So they said, look, why don't we give them a fecal transplant as a Hail Mary to see what it does? So they gave 16 patients an FMT, and half of them responded. They started to respond again to the checkpoint inhibitor that they had become resistant to. So, and they survived. So what it means is that the microbiome is a legitimate target for cancer therapy, as an adjunct to cancer therapy, as is a vehicle for the prevention of cancer therapy. And it's interesting to me that maybe you might even be able to do that through the mouth. Yeah. Hello.
Dr. James, thank you. You were very clever. A lot of it was above my understanding.
Oh, I'm sorry if it was.
I
Yeah.
tried to keep up
Yeah.
there.
Yeah.
The cleft bit really
Yeah.
had me enamoured.
Yeah.
So no one that I spoke to about my kid's cleft mentioned these antibiotics at all.
No.
And in 2017, there was such an influx of cleft children in Surrey.
Yeah.
I had to pull 2 doctors out of retirement to deal with that high level.
Yeah.
They took all of our data for cleft collected.
Yeah.
They're going to analyse it. It'll be ready by the time my kid's like 25.
That,
They
well,
didn't ask me anything about antibiotics.
well, of course they won't. They haven't at all. I mean, so I went to give a... I can tell you that I went to go and speak at their national conference and said, I think you guys need to think about the oral microbiome, both in the causation of cleft palate, but also in its treatment, because I think when you have surgery for the cleft palate, you're reshaping the microbiome. And they sort of looked at me a bit blankly. They were very interested in it, but you have to remember that it's just very far removed from their day-to-day treatment of the condition. And I think there might be lots of other reasons why there was that big spike in cleft palate incidence in
Just a few months.
Yeah.
Had UTI.
Okay.
It's not looking good.
I mean, look, you know, antibiotics are a very precious medicine that I can't do my job without. Like, I've literally just given a bunch of them to a patient that I've just operated on. And I'm not anti-antibiotics. They're also a phenomenally precious resource because we're running out of them. We're resistant to all of them. And, you know, that's a big problem, right? But antibiotics cause chaos. They cause chaos. And my clinic, you know, is full of young people with, you know, if as dentists, really spend some time taking an antibiotic history, and it's just so prevalent. Like my patients with IBS, if you really take a proper antibiotic history, I'm like, were you an earache kid? Oh, yeah. Were you a tonsillitis kid? Oh, yeah. I was like, forever, you know, taking antibiotics for my tonsillitis. And then you sort of start to think, well, hang on a second, you were doing that in your formative years when your gut was developing. No wonder you're in a complete state now. The challenge, the hard bit, is doing something about it. It's unpicking it. It's rewinding it and saying, well, okay, you know, it's very nice for me to be nice and smug and say, well, that's your problem. It's problematic if I can't make them better. And so I would be patient with your cleft palate pals and child because I think they really want to learn and they're trying desperately. And there's some brilliant people in that organisation really trying to make a difference. And actually, this is new science. And it's just going to take a time for it to filter through. Thank you very much. I hope your child's okay.
Well, she's got me as a...
Yeah,
Maybe not.
yeah, I'm sure she's doing just fine. I'm sure she's doing fine, and they're brilliant. Thank you. Any other questions? Yes, ma'am.
It's not so much a question, but a comment,
Yeah.
because I appreciate the harm that antibiotics can have.
Yeah.
And a lot of clinicians are super flippant with how frequently they prescribe it.
Yeah.
And one thing I've been encountering, I'm a specialist orthodontist.
Yeah.
And I see a lot of kids with mouth breathing and things like that. And there's a big connection with ENT. And on the NHS, I've repeatedly seen kids have tonsillitis, and it's
Yep.
7 courses of antibiotics in 1 year for them to qualify for tonsillectomy.
Yeah.
And I've had a kid saying, I only had 6 last year, and so I have to start the count again next year.
Yeah.
Which is absurd.
Yeah.
And
And
I don't know how to communicate it to those people.
well, it's very difficult to communicate it, and part of the challenge is, of course, that child is probably really suffering from that tonsillitis and is really miserable, and you can really understand why their parent wants it and why the doctor wants to prescribe it. That might be an argument for why we should be operating on more tonsillitis. I just don't know. It also raises interesting questions, because I see this problem a lot in the gut. I see it a lot. In fact, I see it a lot with recurrent urinary tract infections. Women with recurrent urinary tract infections, they get stuck in a cycle. Right? It's this classic antibiotic cycle. Like you have a pathogenic problem. You've got a pathogen that's causing, a staph or a strep that's causing an infection. So you take a really broad spectrum antibiotic that might suppress the pathogen for a little bit, but it just wipes out all of the other symbionts in that network that you need to maintain tonsillar health, for example. So what happens? Well, you don't completely kill that pathogen. You might seed, or maybe you create a resistant, variant of it, or maybe actually what grows in its place is more problematic, and then it blooms back again. So what do? You have another course of antibiotics. What does that do? It suppresses what little, mucus you've got left, and you go round, you go round, you go round. So all you can do is communicate and educate and chat and give good nutritional, dietary, microbiome advice. But it's a societal problem. It's a policy problem. Yes. Yeah. No, they're discretely different. They're discretely different. We've been trying to study the microbiome of those for ages, because I think it's quite important, particularly in the kind of rising incidence of NETs, which have gone up 500% in whatever it is, a decade. But yes, but no, because the cell, it's a neuroendocrine cell. Microbes have a very different relationship with enteroendocrine cells in the gut. They're important. It's very poorly studied because NETs are relatively rare. But it's an active area of work. There's a lady called Andrea Frilling at Imperial that's studying it. Sorry, yeah.
So with inflammatory bowel disease, that was really interesting. Hearing about that. So you were mentioning about how antibiotics affecting that thing.
Yeah.
So what would your treatment be? Can you test their microbiome and see what they're lacking and give them specific probiotics for them? Or
So
is it diet or stress?
it's very difficult, is the answer, because what we lack is precise measures of really what's happening in the microbiome to really target it. And there's very limited agreement on what those measures should be, what those analytical tests should be, and how we should interpret them. I've started to use them in my practice. And here's one example. Right, so here is data from, this is long-read sequencing data. The significance of that is I can get down to species level, or substrain level, actually. So this is a phylogenetic, it's called a sunburst pop. That's a bacteria, and each ring is a different taxonomic scale, right? So that's a phyla. So you've got your Bacteroidata and your Bacillota. These are your Firmicutes. Anyway, by the time you get to the outer ring, you've got individual strains that you can actually measure, right? So it's quite useful. So what you can see in this case is that this lady had ulcerative colitis. 2/3 of her gut was made up of Phycocola vulgaris. And that's really problematic because they produce lots of hydrolyses and they produce lots of enzymes that promote inflammation. So the solution when we knew that was not to give her antibiotics, because that wasn't going to solve her problem. The solution was nutrition and diet. It was to work with a dietitian in a very targeted way to try and suppress the population of that. So 2 months later, you can see it's still there. It's down here, okay, but it's gone from being 2/3 of her gut to about being 1/3 of her gut. And she felt better. She got better. Is it causally associated? Hand on heart? I don't know. But I'm now certainly of the opinion that microbiome testing has a place in clinical practice because the microbiome is important in determining a treatment response. The problem is that it's not yet ready for prime time. It's not yet ready for diagnostics. It's not a good diagnostic test, and we should not be using it in that way. And the other problem is that you really have to understand the test to use it correctly, right? And actually, consumers should not be using these tests. They're basically meaningless to consumers, I think. Unless you're kind of really comfortable in understanding them, it's not very helpful. But I do measure them. That's the answer. Yes?
So if
Yeah.
I presume you probably do, but if you had the ear and the budget of the Minister of Health, what would...
God, yeah. definitely got the wrong end of my stick. I definitely don't have the ear of the Minister.
What would you like to see happen?
So what I think is, I can tell you exactly what I would like to see happen. I think my current shtick is that I think the maternal microbiome should be a basic human right. And much like we have a WHO code for kind of breastfeeding, I think that actually we should be moving to protect the maternal microbiome, because if you don't protect it, the infant is then exposed to a lifetime of non-communicable disease risk from which they basically can't escape. So I would like to have policy that enshrines it and protects it. And I think there are ways that you can do that. I think we need to do quite simple things right. So, for example, post-Brexit, we were supposed to have looked at our antibiotic prescribing regulation in farming for animal husbandry, and we haven't done it. And we're behind Europe on that, and we should be doing that. We absolutely should be doing that. And I think our NHS antimicrobial resistance policy, which is quite good, which is trying to reduce antibiotic prescribing, for the right reasons, but for AMR, should include microbiome science in its strategy. And what they have to do for that is to educate. And the people they need to be educating are not consumers or the public. It's basically my colleagues. It's clinicians at all levels, right? Because actually, they I think are still quite mistrusting of the microbiome. I don't think the microbiome has really, reached mainstream clinical practice yet, and it's still seen as a little bit woo-woo.
So all of your microbiome stuff is predominantly in private practice, or are you?
No, it's in the NHS. But I'm quite lucky because I've got a research group, so I can do research. But I can't do microbiome testing for my NHS patients. The NHS won't fund it yet. But again, in fairness to the NHS and in fairness to all sensible clinicians out there in the world, all of the majority of data on the microbiome is experimental. It's not clinical, right? It might be done in humans, but it's not designed as clinical trials to clinical endpoints to build an evidence base, right? And actually, the microbiome science has got to do the work. But at the same time, you've got to give it a little bit of room because it's in baby science. We've only been going for 20 years. We discovered a brand new organ 20 years ago in the gut, and every day we're working out what it does. So I think part of the problem is that the cart has come before the horse. The consumer is very, very engaged on the microbiome. And that's because the microbiome fills a gap in the failure of medical therapy. And it's because social media misrepresents the microbiome and can make it be the solution to any problem that it really wants it to be. And that's what I meant there's a lot of bullshit. Because it's very easy to bullshit on the microbiome because it's kind of overwhelmingly complicated. So I think some skepticism and cynicism is sometimes well placed and well meant because actually you need evidence like that. Let's just be honest about it. Clinicians will change practice when you go, if you use this test, your patients will get better. And so the onus is on us now to create the evidence. Thank you. That was my pleasure. Thank you.